Good morning, student. Today our topic is translation and this is part 5. In my last lecture I discussed translation initiation both in eukaryotic as well as prokaryotic system. Now we will start translation elongation process. Ribosome elongate polypeptide chains in a three-stage reaction cycle that adds amino acid residues to a growing polypeptide C-terminal. So the synthesis is going from 5' to 3' orientation. Now there are three steps you need to remember when we are discussing about chain elongation. Number one is decoding. Number two is transpeptidation. That means the protein synthesis, the covalent bond formation, and the third one is translocation. Decoding. Now what is decoding? It is in which the ribosome select and bind an amino acid tRNA whose anticodon is complementary to the mRNA codon in the A site. Transpeptidation, as I mentioned, in which the peptidyl group on the P site tRNA is transferred to the amino acid group in the A site through the formation of peptide bond. That means the peptide bond formation is taking place. The third is translocation, in which A site and P site tRNAs are respectively transferred to P site and E site accompanied by their bound mRNA. That is, the mRNA together with its base paired tRNA is ratchet through the ribosome by one codon. That means which is now becoming the P site. That P site now become E site. The existing A site will become a P site. A new A site will be generated for the incoming tRNA. Now to understand the process, we will also go and study this three mechanism. Now this left hand side diagram is all three mechanism together, how actually it looks like. So that the first one is what we discuss, decoding. That means the correct amino acid should come and bind from the A side. Okay, so amino acid tRNA bind to an A site. This is the first reaction. Then the peptide bond formation, that is the second step, where the growing chain will be transferred to from the P site to the A site. Now after that, what as I mentioned, there will be translocation. So the P site, former P site, that tRNA will be uncharged. Now no growing chain is there, so that will eventually go to the E site. The existing P site become E site, A site become P site, a new A site will be generated. So this is the way, so that another molecule can come and bind. So you can see one tRNA at the E site, you can see at the P site the growing chain, and A site is vacant. These are the three steps, but if the question comes, you need to discuss in these three steps with the proper mechanism, with the how this entire mechanism is happening with the help of some other translation factor. So these factors also named as elongation factor, EF. Now the first step, that means the decoding. This is done by a translation elongation factor EF, elongation factor TU, thermal unstable. That's the name of this protein. Now, this protein binds with the GTP. It has an affinity towards GTP. Now, where this protein helps to escort the amino acid tRNA, where which side? A side. So there is a factor binding center at the just adjacent to the A site, which is present inside the bigger subunit of ribosome, from where this factor actually enter and occupy the A site of the ribosome. And once it is bound correctly, then there is a GTP hydrolysis takes place. So GTP becomes GDP and PI, and this EFTU will lose its affinity towards this factor binding center of ribosome big subunit. So this is the first step. Now what is the second step? The second step is the peptidyl bond formation. You need to understand one thing, this peptidyl bond formation, this reaction is solely catalyzed by RNA, not any enzymes. So where the RNA are there, the rRNA which is present in the bigger subunit of ribosome, particularly for prokaryotes, 23S rRNA takes part in this peptidyl-transferase reaction. No proteins are involved. So after finding this involvement, that RNA can work as they are functioning as a protein. That's why scientists have interpreted that the ribosome is a ribozyme. Now, once the correctly charged tRNA has been placed in the A site and has rotated into the peptidyl-transferase center, the peptidyl-transferase center present inside the ribosome, peptide bond formation takes place. This reaction is catalyzed by RNA, specifically the 23S rRNA I just mentioned of the large subunit. Early evidence for this came from experiment in which it was shown that a large subunit that has been largely stripped of its protein was still able to carry the peptide bond formation. So the experiment scientists have done, they have constructed a type of ribosome where they are removing one by one protein. As you know, the ribosomes are composed with rRNA as well as ribonucleoproteins. So they are actually removing the proteins, and still that peptidal formation reaction was happening. So the question comes, who is actually taking part? Because we have an idea that always the enzymatic reaction will be done by protein. But when they have done and they have actually gone through the vicinity of the active center, they found that within 18 angstrom there is no protein present, only rRNA, those who are actually catalyzing this reaction. So that's what they mention. Higher resolution 3-dimensional structure of ribosome, which reveals that no amino acid is located. That means there is no trace of protein closer than 18 angstrom from the active site. Okay, because catalysis requires distance in 1 to 3 angstrom range, it is clear that peptidyl-transfer center is the ribozyme, that is an enzyme composed of RNA. So the actual peptide formation reaction is catalyzed by rRNA, which is present in the ribosomal big subunit. For prokaryotes, it is 23S rRNA. No stress of protein amino acids are taking part in this reaction. Now, the third one, once this peptide bond is done, then the third step was translocation. That means the existing P site will be now E site, the existing A site will be now B site, a new A site will be generated, as well as mRNA also need to move with 3 base, one codon ahead. How that can be is happening? So that is done by, so what I have, this picture, what it is showing, that is the third step. This is again the E side is free. P side, you can see the growing change is there, and the new tRNA has come. Now the peptide bond formation reaction takes place. Now the factor here comes EFG, that is also comes with GTP. It comes and bind the factor binding center of bigger subunit of ribosome. But by the time the peptidyl-transferase, that peptide bond formation reaction happened. Once it happened, you can see this thing, there is a conformational change of EFG GTP. So this fellow become, now it hydrolyzes its GTP and release the phosphate and become GDP, and its conformation change which allow it to go down through that A site down to the mRNA sequence, that region. Because of that, it actually acts as a pushing the existing tRNA who was occupying the A site towards P site. So actually pushing the tRNA to go from the A site to P site, and which tRNA is present at the P site, from P site to E site. So after that, once this thing is done, you can see the tRNA which is deacetylate, there's no charged molecule there that is occupying E site, the growing polypeptide chain with the tRNA. It is occupying at the P site, and A site is vacant. Now through this exit point, that other tRNA will get released, and this GDP bound EFG also will get released, leaving the ribosome correctly placed the tRNA with its growing polypeptide chain. So here we can finish the translation elongation process. Surprisingly, this elongation process, the mechanism in prokaryotes as well as in eukaryotes, these are same, but the name of the factors are different. Okay, that EFTU named as EF1, and that is another one is EF2. Now, if I conclude like this entire elongation process, now we can start from this one, where you were seeing that EFTU, we start with this ribosome. This figure, they are not showing the E site. Always remember A site, P site, and E site is also there. But here that EFTU GTP is coming, you know that it is actually escorting the amino acid tRNA, and after GTP hydrolysis, it is getting released from this ribosome, and because of that, the newly charged tRNA has entered. Now, there is one more point I need to explain, that EFTU GDP then again get converted to GTP. Now, this conversion GDP to GTP was done by another factor which is named as GEF. You know that GEF, guanine nucleotide exchange factor. The name of the protein here is EFTS. This is elongation factor thermostable. This takes this GDP and form an EFTU EFTS complex, which again bring the GTP and exchange with GTP with EFTS. So EFTU again will be charged with the GTP and EFTS will be releasing. So EFTS is helping the EFTU to convert from GDP to GTP form. Exchange factor. Now this is the step 1, which is named as decoding. Now the second step is transpeptidation, where it is catalyzed by ribozyme. And then the third one is translocation, which is with the help of EFG. It is again new A site is generating. Yes. Now, throughout the discussion of translation elongation, we have not distinguished between the prokaryotes and eukaryotes, although the eukaryotic factors analog of, that's what I was telling you, EFTU is named as E. E stand for eukaryote, small e, EF1, and EFG stand for, small e stand for eukaryotes, then EF2 are named differently. Their function are remarkably similar to their prokaryotic counterparts. So what is the message here? That eukaryotic and prokaryotic translation elongation process are similar. Only the two proteins EFTU and EFG are named differently, like EEF1 and EEF2 respectively. Now, once the elongation is getting, we are finished. Now the last part of translation mechanism is the termination. Now, there are two class of release factor. The termination, as we know that once this protein synthesis, it will proceed unless until it encounter a stop codon. And you know that there are 3 stop codon. Once the stop codon it encounter and there is no tRNA specified for this particular stop codon, so protein synthesis halt there. Now it needs some helping protein which will help them to get the growing polypeptide chain to be hydrolyzed, and all the factors who are bounded there should get free, as well as ribosome should get free, tRNA should get free, mRNA should get free. So then that is we call as a termination. Now there are two class of the release factor who help to release the protein. One is class 1 and class 2 factors. Class 1 factors, it release factor, they actually recognize the stop codon and hydrolyze the growing polypeptide chain. Remember, class 1 recognize the stop codon and hydrolyze the growing polypeptide chain. But then what is the function of class 2? class 2 factors actually remove the class 1 factor who are actually still there after doing this hydrolysis also, and remove all other factors also eventually. Now in prokaryotes, we have two class 1 release factor. So those are named as RF1, RF2. Now RF1 recognize the stop codon UAG and RF2 recognize the stop codon UGA. But the third stop codon UAA is recognized by both RF1, RF2. Let me tell one more time. There are two class of releasing factor, class 1, class 2. Class 1 function is recognize the stop codon, hydrolyze the growing polypeptide chain. What is the function of class 2? To release the class 1 factor. Now, there are two types of class 1 release factor in prokaryotes, RF1, RF2. Both recognize UAA, but RF1 also recognize UAG, RF2 also can recognize UGA. Now that this is done in eukaryotes, all these three function. What are the three function? Recognition of stop codon, hydrolysis of the proton, hydrolysis of the polypeptide, and then releasing of all the factors out of this entire mechanism process. These three function done by only one protein releasing factor named as eukaryotic releasing factor 1. Small e stand for eukaryotes. Well, fine. With this knowledge, let us start this mechanism. We will first discuss the left hand side, then we will go for the right hand side. Both are important for translation termination. Now, look at this situation. What happened? A growing polypeptide chain is there where at the P side. Now, at the A side, the other one, the another tRNA which doesn't have any charged molecule, which that occupies at the E side. Now, as ribosome encounter a stop codon which is present in the green colored mRNA, then it holds that what to do. At this position, it recognizes now the time has come to stop. Now the releasing factor, there are class 1 factor. Let us see the class 1 RF1. There are two types of factor, RF1, RF2. So let us see that RF1 comes and bind there. Being a protein, it uses its 3 amino acid sequence to recognize the anticodon. What are the 3 amino acid sequence? SPF, serine, proline, phenylalanine. Remember, huh? And there is another domain. There are another 3 amino acid, GGQ. It is not C, it is GGQ. Okay, this is written top, GGQ, G glycine, glutamine. These 6 amino acids are very important. Let me first tell about that SPF. With the help of these 3 amino acids, it recognizes the anticodon, the stop codon. Okay, so this 3 amino acid also named as peptidyl anticodon. So just think like that. This RF1 not structurally but functionally mimic a tRNA. With the help of SPF, serine, proline, phenylalanine, 3 amino acid, it recognizes the stop codon. Okay, it could be UAG or UAA. Okay, whatever it recognizes. And on the other hand, it has another 3 amino acid, GGQ, glycine, glutamine. This interact with the peptidyl transferase center where the peptide bond takes place, that center, and it trigger the hydrolysis of the growing polypeptide chain. So at this point, what we will find that this polypeptide chain get hydrolyzed. Now, second step, the class 2 releasing factor, RF3, it comes and bind. Remember, this is a factor, it comes and bind with GDP, not GTP. Binding of GDP RF3 actually cause the release of this class 1 release factor. So first step, growing protein chain get released. Second step, what happened? This release factor got released. Now RF3, which is now bound to the big subunit of the ribosome, now change GDP to GTP. Now it is GTP RF3. What it does, it releases the release factor 1, class 1 release factor. And now what is there? This structure we are talking. Still two tRNA are there. Still RF3 with GTP is there. Still the mRNA is also there. Now once this is the stage, then RF3 hydrolyzes GTP and it gets released from the ribosome. Now who are still occupying the ribosome? Two tRNA, one mRNA. Now, how that will be released from the ribosome, and how the ribosome as well as the tRNA as well as the mRNA get released from each other. This is done by other one type of protein, ribosome releasing factor, RRF. Here, look at this. Here come another protein, RRF, ribosome releasing factor. So that mimics a tRNA, which also bring EFG-GTP. If you remember the third step of translation elongation, that means translocation, we have already discussed EFG-GTP. It comes and binds to the factor binding site of the ribosome near the A site. So what happened, and then it hydrolyzes and it changes conformation and it goes down. GDP bound EFG is the changed conformation molecule which can go down to the smaller subunit also, okay, and has the tendency to push all the free tRNA to get rid of the ribosome. So exactly the same thing it is doing. After hydrolysis, the tRNA getting released. Then because of this change, there is a conformation change, and these two factor also can next is get released, and then comes the IF3. If you remember initiation factor 3, what was its function? It occupies the exit site of the small subunit, and once it occupied the smaller subunit exit site, E site, the bigger subunit cannot come and bind to this with the smaller subunit. Now you get all the releasing factor done. Both the subunit of the ribosome are separated, mRNA also separated with the help of these releasing factor as well as ribosome releasing factor, RRF, together with EFG-GTP. So this is the mechanism of translation termination. With this, I am finishing today's lecture. Thank you very much.